reason I'm not being able to load to share the screen with you guys. So just give me a second. I'm trying to figure out how can I get the file to display. Okay, it seems like we are getting somewhere. Okay, do you guys see the screen?
We see the screen, but
only the folder, what you open.
Yeah, folder's name.
Oh, you don't see the application?
No,
No, not yet.
no.
Okay. Or let me try to share with you guys somehow. For some reason, I'm having problems here with Zoom. So let me try again.
Maybe you can just restart the Zoom.
Yeah, that's what I'm trying to do, is to restart the Zoom. For some reason, it's not letting me close it. So this is what you guys see on the screen sharing, right? You see directory, right?
Yes.
Yes.
So you don't see the file, right? Okay. Okay. Give me a second. How about now?
It's
Yeah,
working.
recording in progress.
Okay. Thank you. I don't think it's okay. I have to be one of the students, okay? So I guess I'm getting old.
Still young.
Yeah, that's right. Okay. So we stopped, I think, right here when we finished base C, okay? And then with that, I perhaps has covered mainly the majority of the methods, the regression type Bayesian multi-regression analysis for GWAS and genomic selection. The only thing I want to take a few minutes to talk about is the situation when you have non-genotyped animals. Imagine a situation where you have to run a genetic or genomic evaluation, and obviously not all the animals are genotyped, right? You have some animals there in the pedigree, but they died. Some animals you cannot afford to genotype them because it costs a lot of money. So at the time of carrying a genetic evaluation, and if you're going to use this type of model we have been talking about in the last couple of days, then definitely you're going to face a situation where you have an animal, several animals, they are not genotyped. So just envision this in your mind. You're going to fit a model with multiple regressions, except for some animals, those regressions are missing because you don't know the genotypes for the SNPs of the animals that you didn't type or you didn't genotype. So what do you do with that in that scenario? Are you going to run a genetic evaluation just for the genotyped animals? In that case, it's not practical because the industry will not accept that, or you have to do something about that. And that was not an easy task at the beginning, early days of the genomic selection when you use the so-called SNP model or regression analysis. But with time, few approaches have been put together in order to accommodate non-genotyped animals. I'm not going to go too much about the technicality and the derivation associated with these type of things, except to give you a little bit of a flavor about what goes on and what are the implications of considering non-genotyped animals in a genomic evaluation using SNP model or regression models. So basically, if you look at, you have a set of animals that some of them are genotyped and another set is not genotyped. So let's say we have Y1 are the phenotype for the genotype animals, another Y2 vector of non-genotyped animals, and each one of these animals will have breeding values in the vector U1 or U2. So based on that, if I assume the model at the end of this file, I will be running into problem because this Xij, which is the genotype matrix, will not have information for the non-genotyped animal. So what to do about that? You cannot fit a regression that way. So the idea was to basically try to, and it's intuitive, try to, if I don't have those genotypes, then perhaps the best way or the best thing next to having the genotypes would be to impute, meaning to make some inference or some statistical statement about what might be those genotypes if they were observed. The good news is that when we use related individuals, like it's the case in livestock and poultry application, animals in the pedigree are related. And having some genotyped animals that are related to non-genotyped animals, that in itself is a piece of information that could be used in order to infer the missing genotype for non-genotyped individual. And that's the basic idea. How to get it? Well, basically, it consists in partitioning the set of breeding values into two groups and then look at the relationship that exists between the breeding values or even the genomic relationship, the genomic breeding value through the existing pedigree information that we have through the collected data over time and the relationship that exists between the individuals. So basically, one can transform the whole question to a scenario where I have to impute the genotypes of non-genotyped animals, and after a few things like that, using very basic, and I'm quite sure that the vast majority of you are familiar with how to decompose a joint distribution of two random variables that are normally distributed with a known mean and known variance. If you have a joint distribution that is normal with known mean and known variance, deriving the marginal and conditional distribution of each one of the component of that vector U, it's not too much of a complication and it's standard statistical results. So you can write the joint as a product of a marginal distribution and a conditional distribution. And the good news, both distributions are normal, so there is no major statistical computation. And I'm not asking here to understand everything that is on the slide, other than we can write the conditional distribution of non-genotyped animals or the genomic breeding values of non-genotyped animal given the breeding values or the genomic breeding values of the genotyped animals and the pedigree relationship. And through that, we can impute the genotypes of the non-typed individuals. And as you can see here, in this matrix notation, that this quantity A12 times A11 minus 1, and these are all elements either of the relationship matrix itself or the inverse of the relationship matrix times the genotypes on the typed animals. And then I have again a system that is reasonable to implement and is efficient somehow to infer the genotypes for non-typed individuals. And this is the system of equation you will be dealing with. It fits itself within the classical normal set of equation. And we are very familiar with this kind of system, and we have efficient implementation tools to do so. This is the final resulting system of equation. It's basically adding an extra row and an extra row and column to the mixed model equations, and then you do your implementation very similar to what you do in a classical genetic evaluation. The only issue here is that you have to compute some element of A, and then you have to invert some corner of A, and those are not necessarily easily obtained because you have to do some multiplication, and sometimes it's not straightforward computationally to do all those multiplication of matrices and everything, which could be computationally demanding. But other methods and way of implementing this model have been proposed, and right now it's reasonably possible to implement these type of things without too much of computational pain. Okay, so with that, I will stop here. I'm not going to go any further into non-genotyped individual, but just keep in mind that when you have the population of genotyped and non-genotyped individual, and you're going to use a SNP model, in that case, you have to be aware what you're going to do with the non-genotyped animals. Either you discard them from the evaluation, in that case, you have to have a solid argument why you are doing so, or if you're going to include them, you have to use something of this nature where you try to impute the genotype of non-typed individual. With this, I will close the part of implementation of genomic selection or GWAS using SNP model, and I'm going to move to the next other type of procedure we use to implement genomic selection, and it's perhaps the one that is often more used in practical application with large data sets. So I'm gonna close this file, and I'm gonna bring the next file if I don't have any problems, and we'll take it from there, okay? Okay, so now I will be talking about more of a second way of how we implement genomic selection, and it's philosophically a little bit different from what we have been talking about so far. Remember, in all the methods I talked about up to now, we assumed that we're going to fit a regression model or an association model where we put the effect of the SNPs directly into the model. So we have a multiple regression, and the unknown quantity was always the genotype, the effect of the SNP. So remember that sum over all the SNPs that we have in the panel. So those models we have been talking about will provide you with an estimate of the SNP effect. What I'll be talking about now is it's different from the previous one. I will not be estimating the SNP effect first. We will be trying to estimate the whole genomic breeding value. So your model will not include the SNP effect directly, rather it will include the SNP, the genomic breeding value directly. Why is that important? Why this could be an important alternative? Well, first of all, especially for animal and plant breeders, in the majority of the cases, and I'm not saying always, in the majority of the cases, we are interested in the genomic breeding value as a selection tool. So we're not very often interested in the SNPs unless we are trying to dissect the trait. So the effect of a single SNP, it is not often the major goal of a genetic evaluation. In a genetic evaluation, I want to have the breeding values or the genomic breeding values of my animals in order to identify superior ones for generating the next generation. So from that point of view, having a genomic animal model where I get directly the genomic breeding value is kind of attractive to some people. But some people to have just one value for each animal than having estimates of thousands, if not million, of SMP effect. Now, with using this genomic animal model, the dimensionality of the model is not function of the SMPs. The dimensionality of the model is associated with the number of individuals we have in the pedigree. So it's basically on this, when we talk about dimensionality of the model, it's the same as if you are using pedigree information. Contrarily to when you use the SNP model, the dimensionality or the parameter space of the model is the number of SMPs, which is when you have million of SMPs, then you start running into trouble because the dimensionality or the number of unknowns in the model is a function of the number of SNPs. When you use this genomic animal model, the dimensionality of the problem is equal the number of animals you have, and sometimes that number is much smaller than the number of available SNPs and variants. So remember the model we used before when we used the SNP effects. We used this model indicated in the first formula of this slide. So we have the sum of the effect of all the SMPs. So now look at what I'm going to be trying to do, okay? So I'm going to rewrite the first equation in a matrix notation. So the first one was written as a scalar notation. So I take one cow record and I write it that way. So y sub I, let's say, would be the milk yield of one cow, I, and I write it as a multiple regression on the SMP effect. The second equation, I'm taking the milk yield of all the cows that I have in the herd, and that will be my vector y. You see the bold one? That's a vector y. It will be as big as the number of animals I have in my data set. Then here, I keep exactly the same thing as before, except now I have an incidence matrix I in front of that summation over the SNP effects. I didn't do anything. I just moved from a scalar notation to a matrix notation. And then I can write that model, the previous one, the second equation here, I can write it in a much more familiar representation that we as breeders are familiar with. And this representation is the first equation. I have a model, it has an intercept, and then a kind of regression on a set of random quantities I call U and an incidence matrix Z. So Z in this scenario will be the identity, and U will be equal the sum of the SMP effects. So basically here I'm directly trying to put all the effects of all the SNPs weighed by their genotype into one quantity, and I call each one of them as the genotype, the genomic reading value of that animal, and I call it U. So based on this, I can go ahead and solve a classical mixed model equations. In this case, because I didn't put fixed effects and things like that, I can write my system of equation as you see in the first formula. So you have here a matrix of ones, and then this is another matrix out on the upper diagonal. And the only thing, and for those who are familiar with Henderson mixed model equation, this Z prime Z plus sigma squared times G minus 1 is a quantity that is very familiar to a lot of breeders, right? The only difference is that usually when we work with pedigree data, we have, rather than G minus 1, we have A minus 1. So in the presence of pedigree data alone, we compute the additive relationship between individuals based on the average relationships, based on the pedigree, and that will be A. Here, we are modeling the additive relationship between individuals based on their genomic relationship of observed additive relationship. So this is identical to a system of equation you will have if you are applying classical pedigree data, using classical pedigree data, no genomics. If you have no genomics at all, the system of equation you will have is exactly this one, except where you have G minus 1, you replace it by A minus 1. and then you are done. So you see, from a conceptual point of view, breeders are very familiar with this type of model, because since the 80s, we were implementing model exactly like this one, except that rather where there is G-1, we have A-1. That's it. Okay? And this G is the variance of this quantity, which is U, which are the genomic breeding values. And then I can even further go and replace that G-1, which is the variance of the U's, and the U's are the sum of the effect of the SMPs, and you can prove this is equal to G. Okay? So you can see that there is a relationship between what we were doing before and what we are doing now, except that in one case we are modeling the SMP effect, and then we get the genomic breeding value. Now we are modeling the genomic breeding values, and from there we can get our SMP effect if you choose to. Furthermore, I can decompose the variance of the effect of all the SMPs. And I will show that is basically what we proved before, is the sum over the genotype at those SMPs times the variance at each one of those SMPs, which is sigma j, as we talked about in the SMP model. So there is an equivalency between these two models, between either you use SMP effects and then generate the breeding value, or use the breeding value directly in the model, and from there you can get the estimate of the SMPs. Again, with the majority of the cases, even up to today, the majority of the animals in the pedigree, let's say if we take the dairy population in the US, which is a huge population and perhaps the most genotyped population in the world, where we have millions of animals that are genotyped, still, if you look at the pedigree of the Holstein breed in the US, we have tens of millions. So the majority of the animals up to today, they are not genotyped, although we have large number of genotyped animals. For other population, you have just few hundred individuals that are genotyped, and the vast majority are non-genotyped. So trying to implement the model I just presented to you will leave the majority of the animals outside of the genomic evaluation. So what to do? First, we use just the pseudophenotype. So I said, okay, we genotype just the bulls. Let's use the bulls and use the pseudophenotype and move on. But that has a lot of problem, because if you use an estimated breeding value as it was a phenotype, a breeding value has a prediction error variance, so it has an uncertainty attached to it. If you gonna ignore that uncertainty and suddenly assume as if it was an observed trait and model it, it has some statistical risks attached to it. So the idea, and this was Georgia product, was to, by colleagues of mine, is to try to combine a genotype and non-genotyped individual together using the phenotypes and the genomic information at the same time. So basically trying to bring everything together. You bring the pedigree, you bring the genotype, you bring the phenotype and the non-phenotyped and non-genotyped animals. So you take basically everything you have in your population and try to combine them in one single analysis. And this is extremely important practical advantage. Breeders are not statisticians or applied statisticians. Breeders and farmers are producers in the majority of the cases. And what they want is to have a quantity that is as simple as possible to use into carrying their breeding program, rather than worrying about too many variables and things of that nature. So this, at least from practical point of view, is very attractive, because it takes everything, all the information you have, and then you feed it to a one analysis, and you get your genomic breeding values, which you can give to for every animals in the data and the pedigree, and then you can provide it to your reducers. So that's one thing. The second thing is that we have already the infrastructure, the computational infrastructure, largely built from the days of using pedigree-based evaluation. Because these models in spirit are not different from the models we used to have. The only difference is that now, rather than using the additive relationship matrix for every animal, for some animals, we will be using the genomic relationship, and for the animals that are not genotyped, we're going to keep using the additive relationship matrix. That creates a little bit of complexity, how you combine those two pieces of information. Is it clear?
Yes.
Okay,
Yes,
so
yes.
But I
how
have a question.
excuse me?
I have a question. Can I ask?
Yes.
We are using a combination of phenotype, genotype. That's we can call as it is. We are converting G into H matrix.
Not yet. That's where we are going. That's where
Okay.
we are going.
Okay.
Okay. So what I presented before in the animal model would present like assume that all the animals were genotyped. In that case, you have G will cover everybody, right? But now I'm moving to the situation where imagine I'm gonna use a genomic animal model, but some of my animals are not genotyped. Okay? In that case, we're gonna combine the pedigree information with the genomic information, the phenotyped and non-phenotyped individual all together into one single analysis, okay? And that single analysis will have a variance covariance matrix. It's not A-1 as with the pedigree alone. It's not G as with the genomic information alone. It's going to be a matrix H, which blends A and G. Okay? And that will be the final matrix that will be used in implementing this kind of model within a single step analysis. Okay? So this is what we're going to do, and this is I'm going to go over this derivation a little bit, not too much, but just a little bit. So let's assume I have my model, right? The classical model. So U is the vector of genomic breeding values for all the animals. Part of the animals are genotyped, U1. Another group of animals are not genotyped. And so we're going to see now what would be the variance covariance structure of this vector of genomic breeding values of genotyped and non-genotyped individual. Is it clear? So here I'm putting everybody. Every animals I have in the pedigree, genotyped or not, is going to be included in the genomic evaluation. And I'm going to divide this set of individual into one group of animals that have genotype, I call them U1, and a set of animals that are not genotyped, I call them U2. But the analysis is going to be done with all of them. Okay? So again, I'm going to use the same trick I presented to you before in imputing missing genotype for the QTLs. For the SMBs. This is exactly the same thing. I can write the joint distribution of U1 and U2 given the pedigree information A and the genetic variance. And this is the classical assumption of the prior distribution of the breeding values when I have just the pedigree information. This is the multivariate normal distribution prior for the distribution of the breeding values given the pedigree. Now I'm gonna partition that relationship matrix into four corners. You have A11, which are the additive relationship between the genotyped individual. A12 are the individual, the covariance between genotype, non-genotype. Same thing for A21, and then for A22 are the genotype, are the relationship, the additive, the average additive relationship between non-genotyped individuals. Okay? And then now I can right, for the animals that are genotyped in U2, right, also they have, we know that they have another information, which is their genotype. So here I'm presenting the dilemma for you guys. From one hand, I have the pedigree information, which is this information. I have the pedigree information for the genotyped and non-genotyped individual. And from that, I can compute the average relationship between all the animals, genotyped and non-genotyped. And that will be the matrix A. But for the genotyped animals, I have SMP genotype. And with that, I can compute observed or realized additive relationships. Meaning that I can compute additive relationship between the genotyped individual based on the marker information rather than the average relationship. And that will be G. So one will say, okay, what happened now? For these animals that are genotyped, I have two types of relationships. I have the average relationship due to the pedigree, and I have their observed relationship due to the marker genotype I have on those animals. So in naive way, one would say, okay, how about if I just go ahead and take the A22, right? See here from A matrix and replace it with G. And then assume, okay, that's the variance covariance matrix between the genotyped and non-genotyped animals. Anybody can see problem for that? You see what I'm trying to get here, right? I'm saying, okay, because I have two pieces of information for the genotyped animals to compute their additive relationships, one I can do it through the pedigree, the other one I can do it through markers. I'm gonna decide not because the markers are better information, I'm not gonna look at the pedigree information for those genotyped animals and I'm gonna compute their relationship based on the marker, go to that corner of the relationship matrix and replace A22 by G. Is that reasonable?
Yes.
So can we use this and forget about everything else? So why complicating our life and computing a big H matrix and everything? Can I use this matrix?
I think the problem is the covariance, the relationship between it's about A12 and A21. I think.
So what's the problem with that?
I think what's partition we used in this G or effect SNP.
Well, obviously those will never change. Doesn't matter what you do because the non-genotyped animals, the A12 is the covariance between genotyped and non-genotyped. Because you don't have information on non-genotyped individuals, so always it's going to be computed based on the average relationship matrix. So always it's going to be A12 and A21. The only information you have to assess the covariance between genotyped and non-genotyped is going to be the pedigree. For the non-genotyped animals, the only way to compute relationship between them would be through the pedigree. There is something wrong with just trying to put G, take the A22 and replace it with G. And several levels of complexities are there. One is you have two pieces of information. By using what I did here in this slide, basically I said, well, I'm gonna just throw away one of them, which is a waste of information. It's an information that is useful and you decide to just throw it away just because it's convenient to you mathematically. So it's very naive. The second thing, there is no way or no guarantee that you will be able to invert this matrix. That's one, from technical point of view, meaning that this matrix very likely is not going to be positive definite or semi-positive definite, at least. You cannot be able to invert. But even if it has no technical problem, inverting a matrix like this one, It's not going to be possible because it's extremely big. This matrix will be as big as the number of animals in the pedigree. You cannot invert this one directly. Doesn't matter what computer you have or how many years you put, you will not be able to invert. In fact, you will not have any memory to load it before even starting the inversion, okay? So this is extremely naive, and it's not a way to go, okay? What you can do, however, is this, is you go and you look for the conditional distribution of the genotyped versus non-genotyped individual and try basically, it's in spirit, it's not equality in terms, but in spirit, very similar to what we did when we tried to include non-genotyped individual using the SNP model where we imputed their genotype. We're going to use the same trick to try to accumulate non-genotyped animals within the genomic animal model, or the model where we model directly the genomic breeding values. Again, we go to the very basic of decomposing a multivariate normal distribution into a marginal and a conditional distribution. So U is the vector of all the breeding values of genotyped and non-genotyped animals, and I can decompose that into the conditional distribution of the genotyped animals, which are U2, given their genomic relationship matrix, times the genotype of the non-genotyped and of the breeding values of non-genotyped animals, given the genotyped animals and the pedigree. For the first one, U2, which are the genotyped animals given the genotypes, that's a multivariate normal with a known mean and a known variance, and the variance is given by the genomic relationship between those individuals. And then for the non-genotyped individual, this is the conditional distribution for these non-genotyped animal breeding values, which is a mean that is a weighted average of the breeding values of the genotyped animal, and then a variance that is the matrix, the marginal matrix A11 minus this contribution of the related individuals. And then this one, I can put it now into a matrix form and write the joint distribution back. So basically take those two pieces and group them together and create a multivariate normal distribution with a certain mean being 0 and a variance covariance matrix that is given now by a quantity called H. And this H matrix has 4 corners, H11, H12, and so on and so forth. And those elements of this matrix are as listed here. You see in the corner in the second row of the matrix, it's G. That's the variance covariance between the individuals that are genotyped. But if you look at the other terms, they're not similar to the previous matrix I presented to you where it was A11, A12, A22, and G. Now they are much more complex formulas, which accounts for the fact that the genotyped animals and the non-genotyped animals have some relationship through the pedigree. And that thing could be modified just by playing with those expressions over there. You will see that you will end up having a matrix like this. And this is the matrix H I think Amr alluded to. This is what it is. It's a matrix that is basically the inverse of the additive relationship matrix, the average one, A minus 1, plus this contribution. If you look at this contribution, the only non-zero contribution is on the bottom corner of that matrix. And if you look at it deeply, you will see it's a difference between the observed additive relationships due to using markers minus the additive relationship or the average relationship minus the average additive relationship for the genotyped individuals. In A minus 1 is the whole matrix. It will have the 4 corners, right? Including A to 2 minus 1. So we are in A minus 1, we are counting for the contribution of the pedigree to the relationship between the genotyped individual. In that second matrix where we have the zeros and G minus 1 minus A22, we're gonna, J minus 1 will account for the additive relationship due to the markers, but it has to be adjusted for the average relationship due to the pedigree that I already have in A minus 1, okay? So that's why this minus. So basically this H matrix, what it is A minus 1, the average relationship matrix, plus the deviation in the relationship between those due to the genomic relationships and those due to the average relationship through the pedigree, which makes a lot of sense, right? So
Yes.
in extreme case, let's assume for the genotyped animals, the additive relationship estimated through the SNPs and the one through the pedigree are exactly the same. What would be H minus 1 in that case?
A minus 1.
minus 1.
Absolutely, and that's what you expect, right? Because the genomic information didn't bring anything additional to the pedigree information to track the additive relationship between the animals. The more divergence between the genomic information and the pedigree in tracking the additive relationship between genotyped individual, the more diversion will be H minus 1 from A minus 1. Is it clear?
Yes.
Yes.
So that's
Yes.
the advantage of H. Once you get that matrix H, and you see now it's not too complicated, at least conceptually, to see that this matrix is A minus 1, which everybody knows how to compute. And if you don't know how to compute, you can get a subroutine that in few minutes it does you a matrix of million by million. So that's very simple for us. And then everything else, all the computation and the heavy lifting will be related to trying to deal with this extra contribution of the genomic information into the additive relationship between individual. And here I'm not trivializing the thing, right? But it could be very heavy, and in fact it is becoming very heavy. And I'll come to that in a few minutes. But let me just proceed, and I'll come back to talk a little bit about the computational implication implementation problems. But this form, right, of putting the additive relationships between genotyped and non-genotyped animals through this matrix H minus 1 has facilitated enormously the adoption of this type of thing. Okay, so now we're going to have a model, and from here on you can even stop thinking about anything else I will talk about, is that if you remember Henderson mixed model equation, the general ones, right? You can take those same equation, go to that model, every time you find A minus 1, you replace it by H minus 1, and suddenly you become a genomicist. Okay? So we can go even to old notes of Van Black and Dr. Henderson and anybody. Just every time you find A minus 1, you replace it by H minus 1, and then suddenly you are doing genomic selection. Okay? Here are yes.
Please. Yes, about G minus 1, it's SMP effect. Yes.
No.
Yeah.
No.
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 It's genotype also animal genotype effect. No.
No. The G minus 1 is a variance covariance matrix. It's a matrix that will measure the additive relationship between the genotyped individuals using the marker. So we use the marker, and I'll show you, I think there is a few slides a little bit later, how to compute G. G is a variance covariance matrix. It's the same thing as the additive relationship matrix in A, okay? So let me put an example. If you have 5 genotyped animals, each animal is genotyped for 50,000 SNPs. Using those 50,000 SNP for each animal, you try to look at how similar are those SNP across those two those individuals, for every pair of individuals. And that's how you compute those relationships. So an individual with itself or an individual with everybody else, you will be computed based on those marker genotypes. And then the element of G will be elements like, for example, if you look at what is the observed relationship between two full SIPs based on pedigree, it will be 0.5. Based on G, it could be 0.49, 0.51, so something of that nature. No, it's not, they're not the genotype anymore. They are the observed genomic relationship, additive relationship between animals. So the covariance between animals, they're not computed based on the pedigree. They're computed based on the genotype of the SNPs. And that's how the SNP are considered into a genomic animal model.
Okay.
Is it
Okay.
clear?
It's clear. It's clear.
Okay.
Thank you.
So this is the mechanics of implementation. So what I want you to take a look at is just the equation, or the equation where you have P of U given A sigma U square. That will be the only difference from the classical pedigree-based evaluation. That's the only difference, is that when we use only pedigree, the classical evaluations, we have, rather than H, we put A. And A is the expected additive relationship between animals. And we compute based on the pedigree or genealogy file. When you have genomic information, you compute a matrix H. That matrix H will be a combination between the pedigree information and the genomic information. The pedigree information will be used to compute A minus 1. The genomic information will be used to compute G minus 1. You blend those two as I presented in the previous slide, and then suddenly you have your matrix H. You don't need to produce H. If we have to go through constructing H, that would be a major problem. The good news is to solve the system of equation, I do not need H. I need just H minus 1. The inverse, which exactly the equation we obtained here, okay? So H is not needed. What is needed is H minus 1. And that's, we have a very nice formula that we can deal with and we can use in our genomic implementation. Don't have to worry about this. This is how it's done. And you see why I said it's H minus 1. If you look at the last equation, so it involves just H minus 1, does not involve H. And then this one, although it looks a little bit intimidating, but it's not. This is the classical jargonry and tools we use. That's the majority of the programs you run and everything. They use some of these kind of equations to give you the evaluation. I don't expect you to know this, nor to even pay too much attention, other than understand the concept behind what is being implemented. Okay. These are again, the derivations of all the steps to derive all the unknown in the model. This is how it gets implemented using the Gibbs sampler. And here I will spend few seconds, not too much, to give you a little bit of ideas or flavor about this Markov chain Monte Carlo method that are often used in the implementation of this model. Okay, so for those of you who have very little to no information in Bayesian inference, this is just considered as an iterative procedure that uses consecutive sampling from certain set of distributions that are not necessarily the target one, but some intermediary distributions, and then through the consecutive sampling from those distributions, using some of the very rudimentary principle of a Markov chain, namely the first order Markov chains where the future depends just on the immediate past. And that's basically an iterative approach, right? Because we update based on the previous values we get. So you will use a system like this one, and Gibbs sampler is one of the most used algorithm to implement this type of model because it gives a lot of flexibility in dealing with these kind of problems. The only thing I will tell you as the practitioners, if one day you are implementing these models and things like that, that you be very careful when you are assessing convergency of the system and when you are trying to summarize the results, because you could very quickly run into trouble without knowing if you are not familiar a little bit of how these methods work. These are methods that requires huge number of sampling. So you are sampling in hundreds of thousands or sometimes the million of rounds, and you have to be very careful about what you are doing. But other than that, implementing these models are not too problematic from a conceptual point of view. But from practical point of view, there is major problems at all levels, either using the SNP model or using the so-called genomic animal model. And let me take the last one, because the one we have been talking about for the last half hour or so. So when I go back to this matrix, okay, and this is the key matrix we need to implementation of the model, we need to compute the G-1. And G-1 is the inverse of the observed relationship computed through the SMPs. And this is, unfortunately, it's not a trivial question anymore in the reasonable application of genomic selection when you are dealing with a reasonable population. For example, in the dairy cattle population in the US right now, we have several million genotyped individuals. So that this matrix G-1 will have a dimension of few million by few millions. So first of all, you have to compute that 2 million by 2 million matrix, and secondly, and more importantly, inverting that matrix. Obviously, you will agree with me that there is no way on earth you will be able to invert directly a 2 million by 2 million matrix. No, there is no way. I'm not saying that theoretically it is impossible, but it's computationally impossible. You will never have a bigger enough computer or enough time to invert the matrix of that size. And that's not it. In 5-10 years from now, we will have tens of millions of genotyped individuals. So that matrix will keep growing, and it's growing at an exponential pace. Or even some of you with a few thousand individuals, if you are running your evaluation in your laptop, very unlikely you will be able to, and even in a small server, to compute, to store G and inverse, and do the inverse directly. So, although H-1 as presented to you is a very nice and elegant matrix, G-1 is not trivial. Although you don't have as many elements in G-1 as in A-1, A-1 is trivially produced, but G-1 is not when we have large number of genotyped animals. So let me ask a question here. Why it is easy to have A-1 with the inverse of the expected relationship matrix for millions of animals without problem, and we cannot get the inverse of the observed relationship in G when we have few thousand genotyped animals? Anybody knows why? Let me say, first of all, do you guys see the problem? That I could have an inverse, an A-1 of millions by millions without problem, but I cannot have the inverse of G when I have few thousand animals. Any guess? Why is that the case? You get 5 points in the homework or in the final exam.
Can you please repeat the question again, sir?
So why is it that we can invert the average relationship matrix, which is A-1, right? and with millions of animals, tens of millions of animals, without problem. Yet, we cannot invert the G-1 when we have just few thousand animals. Hmm?
It's a trickish one.
How many of you have inverted A-1? Huh? How many
Because
of,
I think it's because we don't need to compute A to inverse it. We can get the inverse of A without computing A.
very good, absolutely. We reduce A-1 without ever constructing A. Set of indirect rules, we are able to construct A-1 without ever. We actually will never construct A to invert. We get the inverse directly without ever constructing A. So that's why we can have A-1 for million of animals without problems. For G-1, it's problematic. It's problematic because you have two problems. The first one, G, the matrix, first of all, you have to compute the matrix G. We do not have indirect rules to compute G-1 like we do A-1. That's one. The second thing, so first you have to compute G, and secondly, and the bad news is that G is dense, meaning there is no zero relationship in G. In G, if you compute the matrix G, and let me show you just one matrix, an example. I hope I didn't erase it. I have one, just for you to give. Yeah, see this, guys? So this is a G matrix between I think 10 animals or so. I just put it together just to show you how it looks like. You don't see any element that is zero, okay? So the matrix G is always very, very dense, meaning there is very few zero elements. So you cannot use sparse matrix techniques to help you in the inversion. This is a compact, very unsparse matrix. And then that makes the inversion, direct inversion of that matrix very, very hard. So if you look at this example, and just I go over a few numbers. So the first diagonal element, 0.9995, that's the relationship of animal number 1 with itself. The second term, 0.4950, is the relationship between animal number 1 and animal number 2. So you can see that that relationship is a first order relationship. It could be either relationship between father or parent offspring, or it could be a relationship between full sips. And it's around 0.5. We talked about why it's the relationship between an individual with itself, or an individual, a parent and offspring could be different from 1 and 0.5, right?
Yeah, right.
So why the relationship of an individual with itself could be different of 1? In absence of inbreeding, okay? There is no inbreeding, yet you compute the genomic relationship like the one I did here, and you have the diagonal element for the first animal is 0.995, and for the second animal is 1.0038. So what's that? How could it be that an animal, in absence of inbreeding, its relationship with itself is different of 1? It's the same reason we used when we explained why the father has a different different than 0.5 relationship with an offspring, okay? Is the finite sampling.
Okay.
It's the finite sampling. So
Okay.
so please, guys, if you don't want to ever forget this, if you have a fair coin, right? Like 25 cents, and I don't know in Kenya, what is the currency in Kenya called? I used to use
Kenyan shilling.
what?
Shilling, Kenyan shilling.
Yeah, Kenyan shilling. I used to have those when I go there to use. So if you take 1 shilling and you start flipping it out and you do it 5 times, right? And you look at heads versus tails, right? So if the coin is fair, what is the expected probability of observing tails?
5.
0.5.
0.5, and for the same thing for heads. But if you do 10 times and you can compute the probability, right? What would be that?
5/10, which is 0.5.
If you do 10 times,
5.
Yes.
or 5 or 10 times. If you flip a coin 5 or 10 times, and then you compute the probability of observing tails, you think you're gonna get 0.5?
No.
I can challenge you. You can bring a shilling and start flipping it out and count. You will not get exactly 0.5.
It's not the 0.5, Prof.
It's not. So why is not?
Because the probability of having a head in every 10 toasting is 0.5 plus the probability of not having the head in every toasting.
Okay.
So just as you have explained the other time, that
Mhm.
infinite sampling of the seeds leads to an individual contributing more than 0.5. So I think it's the same method that might also affect the shilling, either having a head or tail when you toast it about 10 times.
Yeah. So it's just for the small sampling, okay? If you throw this coin 10 times or even 20 times, very unlikely that the probability of observing tails or heads will be close to 0.5. But if you go to the almost infinity, then you will, asymptotically, you will converge toward the data generating process, which is 0.5. This is exactly the same thing. We are sampling few spots in the genome. Although it's 50,000, it seems too large, but we are talking about the genome of 3 billion base pairs. And we are taking 50 locations from that, or benchmarks from that genome. Using those will lead to a small sample of variant or SNPs you are using to estimate the additive relationship matrix. There is an inherited Monte Carlo error associated with your sampling, and that's why you are seeing numbers that are not 1 or 0.5, okay? So it's just due to the limited sampling of the benchmarks you are using to compute these relationships, okay? Now, going back to our question, you cannot invert G because it's extremely dense. There is no zero element in that matrix. And as soon as you have a matrix like this one of 10,000 rows by 10,000 records, meaning you have 10,000 genotyped animals, it becomes impossible computationally to compute that inverse. And that's one of the major bottlenecks we are facing right now in implementation of genomic selection using this genomic animal model, is how to deal or how to generate this matrix H. Remember, this matrix H requires computing G minus 1, right? And to compute G minus 1, you have to inverse the genomic relationship matrix. First, you have to have the genomic relationship matrix and then invert that. Directly, we know it's impossible. You cannot do it now. Unless you have 200 genotyped animals, that's fine. But if you have few thousand or million of animals, then you're not going to be able to do the direct inversion. Then there is some people that are trying to put now approximation, how to approximate the inverse of G, not the direct inversion, but an approximation. One of the methods that is being used is to try to look at, take a set of animals, like 5,000, 10,000 animals as core animals, and try to regress the remaining breeding values of all the other animals that are genotyped on the genomic breeding value of that core set of animals. So basically, you are not computing a complete G-1, but an approximation of G-1. And that's the best we can do right now. And that approximation is obviously data dependent. It depends on how many animals you use in the core, say so many things that sometimes it works very well, other cases it will not work very well. And this is an active area of research where people are trying to figure out ways, and hopefully one of you will step in and find ways, how can we either invert or approximate the inverse of G for large scale implementations. Okay? So yes, if you have small number of animals and things like that, G-1, you can get it, or you can approximate it and you can move on. But as we move along and more genotyped animals are out there, either you use a very crude approximation or you're going to be having problem down the road. Okay? So with that, I will stop for a couple of minutes and then I will take it after that. So I'll stop for 3-5 minutes for if somebody needs to go to the bathroom. I see that there is perhaps few questions in the chat room. Look at them, and then I'll come back and I will be putting few concluding remarks and listening to any of your questions or comments. Okay, so just 3 to 5 minutes.
All right, Prof, while people are trying to ease themselves, I have a question, Prof, please.
Go ahead.
Yeah, concerning the lectures, which your first lectures, which you took us on coding. And, after the coding, where you now told us about that the genomic relationship between two siblings ranges from 0 to 1. Yeah, I was able to get what the relationship is between the other relationships that you also mentioned. You mentioned 3 relationships. I
mentioned what?
You mentioned 3 relationships that exist in genomic relationship. The first one, you said the genomic relationship between two siblings ranges from 0 to 1.
Right.
Am I correct, sir?
Yes.
You also mentioned other relationships, other genomic relationships. So I was not able to pick it up while you were explaining. So I want if you could still explain on that, sir.
Yes. What I think, and correct me if I go out of context, is we were talking about, and the question was, I asked the question, if you do genomic relationships, right? If you compute genomic relationship, like the matrix I just showed you, that the relationship between the parents and an offspring could be different from 0.5. And then, and that's, we try to see why that was the case, and knowing full well that a father or mother will give half of the genetic merit of the progeny. Okay, exactly half. Doesn't matter. It's going to give half of the chromosomes to the progeny. So the relationship between a mother and a progeny, a father and a progeny, independently of what method you use to compute that relationship, should be 0.5. When we use genomic, it's not 0.5 sometimes, and we agree that it's not 0.5 for 2 reasons. One, potentially de novo mutation. So mutation that happened very recently, either in the father or the parents or the offspring, before collecting, let's say, blood sample or hair sample to do DNA sequencing or genotype. That could be, but it's very remote because the number of mutation would be very small to have any meaningful observable effect. The second and the most likely scenario is that the difference we observe are not due to biological reason, are due to statistical reason, to the sampling of finite number of positions along the genome. So you observe those relationships different from 0.5. However, when you take forceps, two brothers, they have an average additive relationship of 0.5. If you look at the pedigree, Yes, the relationship is 0.5. But if you go and you take genomic information and try to compute the additive relationship between two brothers, that relationship could be 0 or could be 1, and any value between those two extremes, okay? And let me put a small example for you, okay? Let's assume the full genome, okay? Let's assume the full genome of a species, right? Just to make things simple, right? Consists of one marker. So the whole genome is one gene, one marker, right?
Yeah.
And let's assume that the father has capital A, little a as genotype for that gene, okay?
Yes.
The mother has capital A, little a for that same gene, okay? So you have two heterozygous parents, right?
Exactly.
So the parent will give the gamete that carry capital A, and the mother gives the gamete that carry the capital A for her first son. So the first son will have a genotype of capital A for that gene, right?
Yes.
The same parent will give, the father will give the gamete that carry little a, and the mother gives the gamete that carry little a for the second brother. So the second brother will have a genotype of little a by little a, okay?
Yeah.
So now if we assume the whole genome is just one gene, if I use these genotypes of the two brothers, right?
Yeah.
Then the covariance between them will be 0, right?
Okay.
If both of them, they inherit the same alleles from the father and the mother, if both of them are capital A, or little a, in that case, the covariance between them would be 1. That's the other extreme. So here I went to a genome that is one marker, right? But in reality, we don't have one marker. We have hundreds of thousands of markers, right? Although in the limit, those relationships could be 0 or 1 theoretically. Obviously, when you have large number of SNPs, you're not gonna see those extremes, but you're gonna see variation around 0.5. Make sense now?
Yeah.
So that's what I was saying, is that if you see different than 0.5 relationship between two full SIPs, it should not be surprising. It could happen using genomic information. But when you see a relationship different of 0.5 between a father and a son using genomic information, the differences are due likely to the sampling of a limited number of positions along the genome. That make sense?
That's good.
Okay. And that applies to other relationships, okay? Full SIPs, half SIPs, you name it, okay?
All right, Prof.
Great.
Yes, Prof.
Any other question, comment? Otherwise, let's go and try to wrap up this thing. Unfortunately, we have 3 sessions. That's all what we have. So I tried my best to put few things together. But anyway, I will try to wrap up this. Perhaps we'll have a few minutes to for a few questions. I have something at 12 o'clock, so I cannot be later than that. So anyway, so this is how we compute the genomic relationship matrix. I'm gonna give you a little bit of flavor about how to compute this matrix G. So basically, we look at the genotypes of the SNPs. For each SNP, for each animals, we look at the genotype. We can code the genotypes -1, 0, 1, depending if it's capital A, little a. And then at each locus, we look at the frequency of those alleles. Say, for example, I use PI as the frequency of the second allele of each marker, and then I can compute a matrix P that is with element equal 2 times PI, which is the frequency of the second allele deviated from 0.5. And then I can create a matrix W, and this matrix W will be the matrix of genotype X minus this matrix P, which basically set the mean allele effect to 0. And then I can use that to compute a genomic relationship matrix. So this is one way how to compute the genomic relationship matrix. So we have this matrix W, W prime, which is the same matrix W over here. This is a very simple matrix to compute because X is the matrix of genotypes that you have it. P is the matrix that have minus to p sub I minus 0.5, and then you compute this. So W prime, this is quadratic form on the matrix W, and then divided is basically, this is standardization that makes the value of in G very similar to the value you see in A, the additive relationship matrix. So divided by the sum of p sub I1 minus p sub I, and p sub I is the frequency or of the second allele of the marker, and 1 minus pi, it will be Q, which is the frequency of the other allele of the marker. And then that will be your genomic relationship matrix. So it's not too complicated. Once there is other ways how to compute the genomic relationship matrix, okay? There is several other ways. So this one is the one often used in our in the field of animal breeding and genetics, and this is what Paul Van Reyden came up with. In human genetics, they use another formula that is not necessarily the same as this one, very related to this one, but it's not exactly this one. So this is how, and somebody asked before the question, how the SNP information is being considered in a, when you use a genomic animal model is through G. So we use the genotype, the X, the value of X, which are the genotype. In the SNP model, we included them as covariate for the SMP effect. Remember, we wrote the sum of X sub ij times beta j. So the X sub ij were the genotype for the SNPs. We include them directly on the association model. When we use the genomic animal model, we use the SMP genotype, the element of X, to compute the genomic relationship matrix. And then the P matrix and this standardization, the denominator we use here, just to make the element of G look similar to the value of A in magnitude and spread. Again, similarly to the A matrix, the average relationship matrix, if once you compute the matrix G of genomic relationship matrix, if you take the diagonal element, you deviate them from 1, what is remaining there is the so-called genomic inbreeding. Okay? Remember, when we use the pedigree, when we compute A, when the diagonal element of A is different of 0, different of 1, why the element of A can of the average relationship matrix could be different of Y? Why? Could it be first? Could the diagonal element of the matrix A be different of 1?
Yes, if there is inbreeding.
Why? So what is your interpretation of a diagonal element in A that is different of 1? So meaning
the parents of this individual has a common ancestor.
So what we call that?
Inbreeding? Consanguinity? Okay.
Consanguinity, it's inbreeding, yes. When the individual is inbred, then in that case, the diagonal element will be different of 1. And that will be 1 plus something. So what is that plus 1 thing? So let's assume, okay, that the parents are half siblings. What will be the inbreeding on the progeny? Can we, if I know that the father and the mother are half siblings?
4.25.
Why is that?
You mean the inbreeding, the inbred individual, or between the two individuals and the relationship between them, their parents, half siblings, correct? So it is 4.25.
Okay, and then so why you say the inbreeding has to be 1.25?
4.25.
What?
4.25.
That will be the inbreeding coefficient?
Yes.
Oh.
You guys agree with him?
Oh, sorry. This is the correlation, the genetic correlation.
Yeah.
Okay. Sorry.
Okay. So anybody else has any guess?
Yes, well, let me understand the question again, Prof, please.
So if you have the two parents are half siblings, what would be the inbreeding coefficient of the progeny of a progeny from those two half siblings mating?
0.125.
Why?
The two parents are half sieves.
Breeding coefficient will
And the
be
half
half
can-
the,
Wow.
sorry.
Go ahead.
The inbreeding coefficient will be half of the relationship coefficient if there is no inbreeding in the previous ancestors.
Very good. The inbreeding coefficient in the progeny is half of the additive relationship between the parents. So if the parents are half sibs, their relationship is 0.25, then the inbreeding on the offspring of that mating would be half of that, it would be 0.125. Okay? So now, if I want, so basically if you compute A for that progeny, the diagonal element for that individual will be 1.125. If you subtract 1, it gives you the inbreeding, okay? The pedigree-based inbreeding. The same thing applies when you use genomic relationship. So you compute the genomic relationship matrix and you look at the diagonal, you deviate those diagonals by 1, and then what remains there is the genomic inbreeding. Obviously, if you get a negative value because the diagonal element is smaller than 1, which it could happen with genomic information, then that inbreeding is 0. Just what you see there is a small error due to the sampling. Okay, this is one of the other ways to compute the, this is what they use in human genetics, so I'm not gonna go over it too much, okay? So this is how it becomes if you have genotypes rather than haplotypes or alleles at the SNPs. If they give you the genotypes coded 0, 1, 2, this is the formula you can use to compute genomic relationship between two individuals. So this formula I can use to compute the relationship between an individual I and K, and it just based on the directly the genotype of those individuals. Again, you could see non-zero relationship with non-blood related individuals. That's why the matrix G is very, very dense. And you see that, why do you see that? If you compute the G matrix, animals without any blood relationship will show non-zero relationship in the G matrix. First of all,
They have,
go ahead.
maybe they have the same SNPs in low Cs, and when you see, after you think, same SNPs, they have the same SNPs in some low Cs.
So they have the same SNP.
The same allele in the SNP, in the locus of a gene, a certain gene.
And where that comes from. So basically you are trying to look, we say we are looking at the additive relationship between individuals, right? That meaning we are tracking that the sharing of alleles between individuals, right, from a common ancestor, right?
Yes.
So if these animals, they do not have any blood relationship, where that similarity is coming from?
But if we talk genomic, there is some traits or some genes that only have 2 or 4 alleles. So in the population, you will find some individual that has some same allele in some low C.
Why is that? That's why I'm trying to go to. Why is that?
Mutation? No?
No.
Do you mean if the genes are found in the individual and they came from the different origin?
Not necessarily. It's much simpler. It's just due to the fact that blood relationship tracks identity by descent. However, as I don't know who was the first person who answered the question, is that there is another identity with identity by state. So 2 animals will share the same alleles at one position of the genome, not due to identity by descent, not because they inherited from a common ancestor, but it happens to be segregating in the population. And that creates that relationship that 2 individuals, they have the same genotype for a SNP, not because the alleles they have on those positions were inherited from a common ancestor, rather that those alleles are existing in the population And they are, obviously, all of it goes to whatever you believe in, where we come from. And then obviously, if you look deep enough, everything is IBD, right? If you look deep enough, and independently of what you believe in evolution or creation, whatever, then you will see that everything is IBD. But there is some IBD is identity by descent that is extremely ancient, that it basically removes any family relationship in our understanding today. And we call them identity by state. It happens that there is some alleles that are segregated at a certain position of the genome, and some animal will share that kind of those alleles without us knowing of recent family relationship, which indicates identity by descent.
I
So
think that's
always,
this is the similar genes, yeah? Like
it's the same one.
called similar genes.
Yes.
It's not identical genes, yeah?
Right.
If
So
the
let's
gene-
assume,
if
go ahead.
yes. I mean, if the genes inheritance from the different origin, they said it's similar genes. But if they inherit from the same origin, they said identical genes, correct?
Well, there is no different origins. The origins are the same, except that if they are coming from a common ancestor or not. If they are coming from common ancestor, we call IBD inherited genes or alleles. Otherwise, there will be identity by state, meaning that they happen to be because they existed long enough in the population. Obviously, they have been IBD at some point, because doesn't matter. Let's assume if you take the model of we come all of us from one genome, right? And that applies to both philosophy, the evolutionary one and the creational one. Okay, let's assume we all come from one individual, right? If all of us we are coming from one individual, then every gene that is, or every allele that is segregated in the human population comes from a common ancestor. In that case, the identity by state is nothing more than a very ancient identity by descent. But let's assume from our first human, we have, let's say that human is homozygous at a certain position of the genome. And that thing didn't observe major mutation over time, then two individuals could share the same genotype at that position, not due to a very recent family relationship, meaning that inheriting that allele from a common ancestor, but rather because it is existed and it has been segregating in the human genome since the first individuals. And that's what we call identity by state. And that's why every time you take two individuals and try to look at their relationship based on a large number of markers, you will find that those two individuals will share some markers, not necessarily due to their recent family relationship, rather than to inherit some position due to identity by state, okay? And that's basically one of the reasons why we are having problem with G, is that due to this fact, every element in the matrix G has non-zero element. And when it has non-zero element, we don't know if that non-zero element due to a very ancient identity by descent, or is it due to identity by state, or is it due to noise? If we can differentiate the three sources where the relationship comes from, that I think could help us quite a bit in simplifying the structure of G, and then perhaps down the road, the inversion. I'm raising this question not because I'm too concerned about the relationship of 0.002. Unfortunately, that relationship is that its presence in G makes the matrix extremely dense. And by being very dense, it precludes us from doing simplifying the inversion. So if those relationships are true, we need to keep them. But if they are not true, they are due either to identity by state or just to Monte Carlo noise, removing them could make the matrix a little bit more sparse, which could down the road facilitate its inversion. Okay? I think, guys, I have to stop right here. I have another meeting coming after now. So just if there is any burning question or comment, and then I will stop. So if any of you has a question or comment, and otherwise we will conclude with this. Any extra question, comment? Well, if not, thank you for participating. I wished we didn't have COVID, and it could have been a much better scenario. I tried to do my best in 3 sessions to present what I thought is the most important parts of genomic selection. I have a lot of material that I didn't cover, a lot of other material I didn't send, but anyway, it is what it is. I hope it's just a very short general primer for genomic selection, and if any of you is interested, I will be more than happy to answer questions via email, obviously, as I can. And then wish you good luck, and that's it. Thank you for being part of the course, and
Thank you.
thank you for my
Thank you.
email.
Thank you.
Yeah, okay, guys.
Recording stopped.
Thank you, Prof.
There is a question.
Thank you, Prof. I appreciate you.